Um, Good afternoon, everyone. My name is Sing-Hee Lee from Seoul National University. And first of all, thank you for inviting me here today. So instead of discussing the detailed mechanisms of the stem cells, I'd like to show how we are utilizing stem cells for studying the embryonic development, especially for the motor neuron generation. The spinal cord injuries are one of the most devastating injuries in modern societies, and motor neuron diseases listed here are equally frequent and destructive. And one of the most infamous motor neuron disease is ALS, or amyotrophic lateral sclerosis. And another example is multiple sclerosis, which is caused by the damage to the myelin sheath, the protective covering that surrounds the nerve cells. So the treatment for both spinal cord injuries and motor neuron diseases is very limited, mainly because spinal neurons do not regenerate. But the new hope is to differentiate embryonic stem cells to specific spinal neurons and apply them to the damaged areas of the patient. However, the success of this approach requires more complete understanding of neuronal development processes in embryos. The spinal cord neuronal system can serve as an excellent model system to study the molecular mechanisms of many transcription factors and extracellular signaling molecules. And this is the dorsal view of an early stage chick embryo, and this is the transverse section of the spinal cord. Spinal cord is a very symmetric structure, and this diagram shows three signaling molecules that pattern the dorsal-ventral axis of the spinal cord: BMP, Sonic hedgehog, and retinoic acid. So our research has been focused on this, how this PMN for progenitor domain for motor neurons give rise to the motor neurons. In particular, the molecular mechanisms of how extracellular signaling molecules such as retinoic acid and Sonic hedgehog cooperate with transcription factors to specify the motor neuron identity. So at a later stage, when motor neurons are generated from the neural stem cells, the mature motor neurons migrate to the lateral side of the spinal horn. And each motor neurons are segregated into specific subtypes, columnar subtypes, establishing the neural circuit formation for the locomotion control. So for example, the motor neurons that innervate the limb muscles are contained within these lateral motor column regions, or LMC regions. And these LMC regions are produced specifically at brachial and lumbar levels of the spinal cord, whereas medial motor column, or MMC neurons, are produced all the way through the spinal cord from the cervical to sacral areas of the spinal cord. And these MMC neurons innervate the epaxial muscles. Okay. So during the motor neuron differentiation, the Sonic hedgehog, which is released from the notochord and floor plate, activates downstream GLI transcription factors, leading to the formation of distinct classes of progenitor domains in the ventral side of the spinal cord. And when the motor neuron stem cells exit the cell cycle, there are two key players expressed. They are limb homeodomain transcription factors, ALID1 and LHX3. So they have limb domains for protein-protein interactions and a homeodomain for DNA binding. The knockout of these genes results in defects in the motor neuron differentiation. So this slide summarizes our previous findings that briefly, the ALID1 and LHX3 interacts with each other, and there's another protein called NLI, which has a dimerization domain that recruits another set of ALID1 and LHX3, thereby forming a hexameric complex. And we named it motor neuron hexamer, or MN hexamer. And we also reported that this MN hexamer functions through binding to these AT-rich DNA sequences, and we named it hexamer response element, or hexali. So given a potent activity of this MN hexamer in inducing the motor neuron differentiation, so we established an inducible embryonic stem cells that express isletone LHX3 fusion proteins by treating doxycycline. So we utilize this motor neuron differentiation conditions. So basically we make the cell aggregate and treat retinoic acid to promote the neurogenesis, and then treated the retinoic acid together with doxycycline to induce the expression of isletone LHX3 fusion proteins. So we found that this method is more specific and very efficient in driving motor neuron generation than the conventional method using the Sony Hedgehog and retinoic acid. So based on the previous results, we hypothesized that this MN hexamer functions as a master regulator for motor neuron differentiations. And we also hypothesized that this MN hexamer directly regulates a battery of motor neuron genes which has this hexamer response element. And those genes might be involved with the neuronal subtype specification, cell body migration, exon guidances, and so on. So to identify the direct target genes of MN hexamer, we performed ChIP-seq and RNA-seq experiments using the inducible embryonic stem cell systems. And we identified about 500 direct target genes, and right now we are focusing on to figure out the function of each of the target genes during the motor neuron differentiations. So today I'm going to talk about this gene called ARH-GAP36, which is a Rho GTPase activating protein 36. So it was identified as a positive regulator of a sonic hedgehog pathway, and it has been recently published that this ARH-GAP36 blocks the protein kinase A activity and targets pKa for protein degradation. Okay. So I'm going to talk about its critical roles as a signal transduction mediator of sonic hedgehog pathway in the lateral motor columnar specification during motor neuron differentiation. So from the ChIP-seq, we identified the binding peak of MN hexamer binding sites in the promoter regions of ARH-GAP36. So we generated a reporter construct which has GFP or luciferase linked to the 2 copies of genomic fragments which has this hexamer response element. So in the luciferase assays, we found that this reporter was strongly activated by co-expression of isletone and LHX3. And then we examined whether this ARH-GAP36 enhancer is activated in vivo. So we utilized the in vivo electroporation system. So we inject the reporter gene into the chimp neural tube and let them grow for a couple of days and examine where the GFP is expressed. So when the GFP is alone expressed, now we can see very specific expression of this GFP in the motor neuron areas marked by the motor neuron gene HB9. And then we co-expressed isletone and LHX3 together with the GFP. Now we can see very atopic expression of GFP in the dorsal side of the spinal cord. So these data suggest that the isletone LHX3 complex directly binds to these hexamer response elements and turns on its expression in developing spinal cord. And we also found that the expression of ARH-GAP36 was induced in the motor neuron differentiation condition using the embryonic stem cells. As you can see here, it's highly expressed at the mature motor neuron stage. And then next, we checked whether ARH-GAP36 is indeed expressed in the developing spinal cord. So these are mouse embryos, and we found that ARH-GAP36 begins to be expressed in the newborn motor neurons around 9.5, and it was strongly induced around 10.5. And interestingly, we found that the expression of ARH-GAP36 was restricted to the lateral motor column areas at the brachial and lumbar levels, but it was not expressed in the thoracic levels. And POXP1 is one of the markers for the LMC neurons, and all the POXP1 positive cells are ARHGF36 positive. So to figure out the function of ARHGF36 in the developing spinal cord, we atopically expressed ARHGF36 in the chin neurotube. So we can see the nice expression of this ARHGF36. And apparently, the size of the spinal cord injected with this ARHGF36 was increased. And we checked the motor neuron genes such as HB9, ILID1 and 2, and BECT, and all of those are highly strongly induced by ARHGF36. And also, the marker genes for the motor neuron, the ventral progenitor domains, NKX2.2, oligo2 are also atopically expressed in the dorsal side of the spinal cord. So these effects of ARHGF36 is very similar to that of the Sony-Hedgehog pathway activation. So we checked the Sony-Hedgehog downstream target genes such as PAST1 and GLI1, and those are nicely induced in the dorsal side of the spinal cord. Okay, so these results indicates that the atopic expression of ARHGF36 results in a strong ventralization, which mimics the Sony-Hedgehog pathway activation. So to further figure out whether ARHGF36 functions through Sony-Hedgehog pathway activation, we monitored the GLI3 processing in the cells. So in the absence of Sony-Hedgehog, PKA is known to phosphorylate GLI transcription factors, leads to produce the repressor form of GLI. So we confirmed that in the presence of PKA, there's an increase of GLI3 repressor form, and this increase was blunted by overexpressing ARHGF36. And then we tested whether ARHGF36 inhibits the kinase activity of PKA. So wild-type PKA shows the increase of the phosphorylated CRAB, which is a direct target of PKA, and also it increased the phosphorylated serum levels of the putative substrates. But the kinase dead mutant did not show the increase of the phosphorylation. And this phosphorylation was severely inhibited by co-expressing of ARHGF36. And these results also shown in the luciferase assay using the Cre luciferase. And this Cre can be activated by the phosphorylated CRAB. So in the presence of PKA, we can see the nice activation, but this activity was blocked by the expression of ARHGF36. So our results also confirm the previous reports that says that ARHGF36 inhibits the PKA kinase activity. So in turn, it activates the Sony-Hedgehog pathway. So given a potent activity of ARHGF36 in ventralization and motor neuron gene induction in the spinal cord, we hypothesized that whether ARHGF36 alone might be sufficient to induce the motor neurons from embryonic stem cells. So again, we established the inducible embryonic stem cells so we can control the expression of ARHGF36 by treating doxycycline. So we used the motor neuron differentiation assay here, and as a positive control, we treated retinoic acid together with a Sony-Hedgehog agonist. So in this context, when these embryonic stem cells were treated with retinoic acid and Sony-Hedgehog agonist, we can see the nice induction of motor neuron genes here. But when retinoic acid and doxycycline was treated without the Sony-Hedgehog agonist, we can see the induction of TJ, which is the neuronal markers, but these cells fail to express the motor neuron genes HB9, suggesting that ARHGF36 alone is not sufficient to induce motor neurons from embryonic stem cells. And also it says that Sony Hedgehog ligand might be needed for ARH-GAP36 to function properly in vivo. And based on the specific expression pattern of ARH-GAP36 in the lateral motor column areas at later developmental stages, we consider the possibility that ARH-GAP36 might affect the formation of LMC specifications. So we atopically expressed ARH-GAP36 in the ventral side of the spinal cord and harvest the embryos at later stages when motor columns are specified. So interestingly, we found that the LMC marker POXP1 was significantly induced by ARH-GAP36, but there was no effect on other motor columns such as the MMC motor columns, which express LHX3 together with HB9. So these results indicate that ARH-GAP36 is sufficient to direct the LMC fate determination. So we identified that ARH-GAP36 requires Sony Hedgehog ligand to function, and ARH-GAP36 directs the LMC specification. So we considered the possibility whether Sony Hedgehog is expressed in the lateral motor column areas of the spinal cord at later developmental stages. So indeed, at earlier stage, we can detect the Sony Hedgehog only in the floor plate inside the spinal cord. But at later stages, now we can see the specific expression of the Sony Hedgehog in the lateral side of the spinal cord, specifically at brachial and lumbar levels, but not at the thoracic levels. So we considered the possibility whether Sony Hedgehog regulates the LMC columnar specification. So we knocked down Sony Hedgehog in the thick spinal cord. So we confirmed the reduction of the expression of Sony Hedgehog here. And then we found that POXP1 positive and ILID1 positive LMC neurons are significantly reduced, about 50% reduction compared to the uninjected control side here. But there was no change in the LHX3 positive MMC columns. So Sony Hedgehog, which is expressed specifically in the LMC neurons, is necessary for LMC specification. So to further figure out the function of ARH-GAP36 during the embryonic development, we generated a knockout mouse for ARH-GAP36. So, but at older stage, like in 9.5 through 11.5, we could not see obvious defects in the motor neuron formation. But interestingly, from E13.5, we could see the dramatic reduction of POXP1 positive and LDH2 positive neurons, which are the LMC markers. Those are significantly reduced here. But there was no change in MMC columns here, and also other brachial interneurons are not affected. And these are brachial levels. And we also checked the thoracic levels, and at the thoracic levels, there are other motor columns such as PGC, HMC columns, and those columns are not affected by, in the absence of the ARH-GAP36. So from these mouse results, we confirmed that ARH-GAP36 is specifically required for LMC formation in mice. So to fully understand the nature of the ARH-GAP36, we tried to identify the signaling pathways that regulate the activity of ARH-GAP36, so, such as post-translational modifications, including phosphorylation. So we could identify several predicted phosphorylation sites based on the protein sequences, and AKT was one of the high-length kinases. So we searched researchers whether AKT is involved with the Sony Hedgehog pathway, and indeed, it was shown that Sony Hedgehog can activate AKT kinase, and this activated AKT de-represses GLI activity by inhibiting PK activity. So we hypothesized that ARHGAP36 might function as a downstream effector of this AKT kinase in repressing PKA and de-represses GLI transcription factors. So we tested whether AKT affects the ARHGAP36. So we transfected ARHGAP36 together with AKT in HEK cells. So interestingly, we found that wild type and constitutively active form of AKT strongly stabilized the protein levels of ARHGAP36, but the dominant negative form of AKT does not show this effect. And the stabilization of ARHGAP36 by AKT wild type was inhibited by treating AKT inhibitor, but it does not affect the constitutively active form here. And in the co-IP assay, we found that AKT interacts with ARHGAP36, and this interaction was blunted by AKT inhibitor. So, activated AKT interacts with ARHGAP36 and stabilizes the protein levels of this ARHGAP36, which in turn blocks the PKA activity and de-represses GLI transcription factors. Okay, so next we tested whether AKT is required to modulate the Sony-Hedgehog pathway in LMC specification. So we injected AKT into the chick neural tube and monitored the expression levels of LMC marker genes. So wild type and constitutively active form of AKT highly induced the expression of POXP1, but the dominant negative form of AKT further represses the expression of endogenous POXP1, suggesting that AKT is required for LMC formation in developing spinal cord. And this dominant negative form of AKT further blocked the activity of ARHGAP36 in inducing the POXP1 upregulation in the chick spinal cord, suggesting that this AKT is required for ARHGAP36 to modulate the Sony-Hedgehog pathway in LMC specification. So, in summary, I speak quite fast. So we identified ARHGAP36 as a direct target gene of MN hexamer, which is the master regulator for the motor neuron differentiations. And we identified that the Sony-Hedgehog is expressed specifically in the LMC neurons at later developmental stages. So while the role of Sony-Hedgehog in inducing the ventral progenitor domains has been relatively well studied, the function of Sony-Hedgehog in LMC formation has not been studied at all. So we found that Sony-Hedgehog activates AKT, and this activated AKT regulates the protein levels of ARHGAP36, and which in turn blocks the PK activity and increases the active form of GLI, and it might regulate the Sony-Hedgehog target genes. And we still don't know what target genes are related to the LMC formation, but we found that in ARHGAP36 knockout mice, we could see the increase of apoptotic cells. So maybe this pathway is involved with the motor neuron or LMC cell survivors at later developmental stages. Okay, so I'm gonna stop here, and I'd like to thank my lab members. So Hijin Nam, a graduate student, she did almost all those work, and I'd like to thank my collaborators at OHSU. So I'm gonna stop here, and I'm gonna answer any questions if you have. Thank you.